Hello and welcome to our Lakeside Chat. We are David Wofford and Katie Noonan, co-chairs of the Rotary Nature Center Friends. And we're so happy that you can join us tonight.
As we begin our program, it is important for us to acknowledge that we are on stolen land, that Lake Merritt is part of Ohlone territory. We hear now from Corinna Gould, spokesperson for the Confederated Villages of LaShawn and co-founder of Segurate Land Trust.
Good afternoon, relatives. My name is Corinna Gould. I am the spokesperson for the Confederated Villages of LaShawn. We are here today at what most folks think of as Lake Merritt. We are in the territory of Huichin. Huichin is actually a territory that encompasses 6 Bay Area cities: Oakland, Berkeley, Alameda, Emeryville, Albany, and Piedmont. And so this is a place that my ancestors have been since the beginning of time. And this was a place of abundance. I'm so happy that people from all walks of life that now come into our territory can enjoy this beautiful place that my ancestors have enjoyed since the beginning of time. My relationship to the land that I have been born to, raising my children and grandchildren here, has been to tell the story, the truth of what happened on this land before other people came here. I'm hoping that as we begin to learn these lessons of fires in California, the pandemic that's happening, that human beings come back to living in reciprocity with the earth.
Tonight, we have a very special honor. We're going to be hearing from Sean Sommeran, who is the founder and the executive director of the Pelagic Shark Research Foundation. And I've been wanting, ever since I met him back in 2017, mostly online, when we had a major bat ray die-off at Lake Merritt. I've always wanted to have him come and give a talk to our citizens about his important conservation work with sharks. So Sean is a very busy guy, and he's doing all kinds of important work. So I wanted to give you a flavor of this with a short video clip. Are we ready with that, Rob?
She said there was a ray stuck in a slough by 5 Port Royal Avenue in San Carlos, San Mateo.
I'm calling to report a shark in distress at New Brighton Beach. I guess it was on the beach, but I did see a distressed stingray, or it might be possibly dead. I'm not sure. It was in Tiburon,
The Marine Mammal Center. We got a report of a dead shark on the north end of Sunset Beach in Watsonville. Just wanted to let you know we do have photos and can pass on the RP information.
With the Marine Mammal Center in Sausalito. We just got a call about a baby, it looks like a white shark, but I can't quite tell, that has hauled out at Morro Strand State Beach. The RP reported that it may have had an injury of some kind on it. I do have a photo that I can forward along to you guys. But like I said, it was at Morro Strand State Beach in San Luis Obispo. Thank you.
We have a report of a shark that keeps beaching itself at Marshall's Beach in San Francisco.
Hey, Sean, this is lifeguard at Ocean Beach, San Francisco.
All right. So I just wanted everyone to see how busy things can get for Sean as he responds to reports and deals with them and gets calls from Marine Mammal Center and researchers and citizens all up and down the coast. So Sean, I just pulled out a list of some of the species that were on your website, and they're listed here. And then we have some video just came in, so I think we're going to go to that pretty quickly. Can you tell us just a little bit? Here's the picture of the bat ray stranding at Lake Merritt that kind of was the first inkling that many of us had that this was a problem. And many of us have not learned a whole lot more since then, so we're really looking forward to hearing from you. We've got leopard sharks and bat rays. And I think, and this was a blue shark stranding that was on your website. So Rob had 2 really short clips of a bat ray and a tiger shark that was sent by Dr. Okihiro, and bat ray was taken by Adrian Cotter locally. And just to give a picture, just a view of these animals happy in their native habitat, unstranded. Are we ready for that, Rob? It's like, yeah, all right. beautiful leopard shark here, sent to us by Dr. Okihira. Okay. Oh, we're gonna stop at Itchy Scratchy. And then there's the one of the bat ray, which is like 16 seconds. Yeah, it's taken by a local naturalist and photographer, Adrian Cotter. Lots of people here like to go to the bridge and look for bat rays, and they're quite often rewarded by doing that. Yes, this is Lake Merritt. Yeah. All right. Well, Sean, thank you so much for this for taking this long introduction, and I'd like to turn it over to you to talk to us about your work and what a little bit about the species that you're interested in.
Hi, Katie, and thank you, Katie and Rob and the rest of the crew. So I think the most signature species for San Francisco Bay would be the leopard shark, you know, and increasingly the bat rays, because they're so evident, they're graceful and they're neat looking. But there are other species as well, maybe less obvious. The shovel-nosed guitarfish, thorny-back rays, the round ray. We have an electric ray, the torpedo ray in San Francisco Bay, as well as the seven-gill shark. You know, as a note earlier, there was a bit of a discussion about whether white sharks, you know, what's the largest shark that comes into San Francisco Bay? And that would include the white shark, in addition to seven gills. And I've heard some compelling evidence regarding six-gill sharks at certain times entering San Francisco Bay, you know, being relegated and associated with the deeper part of the marine canyon. The seven-gill sharks, especially the juveniles, will come in very close, especially during the springtime into shallow water, etc. And you can do those. Many people see them and just assume that they're leopard sharks, which are the most common and abundant. There's smooth hounds, you know, gray smooth hounds, periodically, you know, the brown smooth hound as well, and the soup fin shark, you know, thresher sharks. There's all kinds of sharks, including, you know, pelagic species that will come into the bay. Thresher sharks have been collected. We've collected a number of thresher sharks there in the past, you know, decade. It appears to be sort of increasing. We've been doing this for 30 years. It was predominantly bat rays, leopard sharks, and or salmon sharks along the coast, and sometimes in San Francisco Bay. So there's quite a bit of overlap, you know, with the coastal offshore species, as well as the estuarine, you know, coastal species, ground species like leopard sharks, etc. You know, California's tidal marine estuaries have been diminished by something like 90%, right? So we have 10% remaining tidal marine estuaries. We hear a lot of talk about, you know, salt marsh, the importance of that, and wetlands. And I wish there's more discussion and concern for the remaining tidal marine estuaries. The people involved in tidal marine estuaries are very much invested and aware and alert, but, you know, it's not a competition, but, you know, with all the other issues that come up periodically, most of the issue familiarity is provided by the, you know, the media, the press, and, you know, there's just not enough media and press to go around for everything. So little, you know, ad hoc, you know, grassroots, homegrown, groundswell efforts like this are really valuable to issue familiarity and situational awareness. In this case, regarding stranded sharks and rays and or fish, you know, including things like giant squid, right? Every once in a while, something washes up that's really, you know, unique and irregular, you know, and that's a value, you know, to the labs as well. Keeping long-term monitoring data on, you know, the persistence of strandings and the composition of the species involved, you know, the environment, the habitat. And I think the strongest asset to that effort is the public, right? There's not a lot of, you know, dispatch or coordination or information on what to and where to report such events, right? So Marine Mammal Center is kind of the magnet for all that kind of stuff. You know, they're task saturated and doing super good work with all the marine mammal, you know, efforts. Everything from recuperating to, you know, whale strandings, you know, and responding to distressed, you know, wildlife, sea lions, harbor seals, otters. You know, all this stuff is super important. You know, but they can't do it all, right? So, but they do have I think the best established stranding reporting network, and they dispatch this information out. When they get reports of a stranded shark, you know, they're not going to necessarily be able to deal with it. They help all the time. But when, you know, oftentimes, you know, they don't have any available assets other than their dispatch, and they will forward these reports to myself, the Pelagic Shark Research Foundation, for going on 30 years now. And I will then share that information foremost with, you know, Dr. Akihiro at the California Department of Fish and Game. It's the right place. Fish and Wildlife Department nowadays it's termed. And he's the perfect guy for that. He's the hub of this effort in terms of, you know, assessing the reports, identifying the species, and then coordinating and organizing a response, whether it's recovery, you know, rescue in some occasions, or possibility. But for the most part, it's kind of gathering evidence, gathering the specimen, you know, fresh as possible, and then shipping it to a lab that knows what to do, what to look for. In this case, Fish and Wildlife Department has, you know, an excellent lab for that, the Cox Lab, Fish and Wildlife Department, and Dr. Mark Akihiro, the chief fish pathologist for that. So oftentimes people make a report to the state parks, the lifeguards, Marine Mammal Center in particular. They will contact us. I will contact Fish and Wildlife Department or the nearest park ranger, lifeguard, to, you know, secure the specimen, right? People sometimes, oh, look, you know, that's a fish I want, I'll take home. You know, it's edible and it looks in perfect condition. You know, oftentimes there'll be, you know, a toxin involved or a pathogen or parasite type of situation. And, you know, maybe you don't want to eat that. Since 2011, bat rays, leopard sharks, and some other species have been, you know, noted as being heavily loaded with bioaccumulative toxins, everything from mercury, PCBs, trace organochlorine, stuff that you don't want to ingest on purpose, or, you know, come to terms with that fact. So a lot of these sharks and rays in San Francisco Bay aren't necessarily good table fare. Some areas are better than others. But people need to be aware. They're not aware of that, for example. Leopard sharks, bat rays, all these different species of sharks in San Francisco Bay are, you know, kind of a favored target for, you know, sport anglers, you know, people foraging for food, which is you know, kind of a big part of fishing, right? People fish for food, or just, you know, sport fishing. So we get a significant number of strandings that are, you know, hook injury. You know, guys catch them by accident. They want to, you know, let it loose. Oh, sorry, you know, let it go. And there's hook damage. It'll wash up not far away, not long afterwards, dead. There is a you know, kind of a growing interest among some sport fishermen. You just, you know, catch them for fun. You know, you go out there on a weekend, you work all week, you know, hard, you get away, you get back to nature, and you want to hunt and gather, and, you know, you don't want to eat them, but, you know, they're fun to catch, right? You fight them. And a lot of people will, you know, mention, oh, we only do catch and release. We release everything unharmed, which is a misnomer at best, right? You know, you're hooking them. If you're using a big barbed hook, it does all kinds of damage to the mandibles, the jaws, there's arteries, nerves, and they do a lot of damage. So you should, if you're recreationally catching and releasing animals, use barbed hook, or barbless hook. You know, it's like salmon fishing, you know, it's for a reason. You know, but we get a lot of strandings that are like that, you know, and then we get, you know, things die naturally, of natural mortality, natural causes. They'll wash up periodically. And then we have this other type of situation where we'll have hundreds, thousands, several or to 10,000 of dead sharks, rays, equal numbers of striped bass, some sturgeon, 7-gill sharks mixed into it. What to do, what to think about that, right? So the best step is to, report that, find the specimens when possible and they're fresh and good condition, ship them to a pathologist, a lab scientist like Mark Akihiro and all of his associates, where they can properly work it up, without so much guesswork, right? They know where to look, for these pathogens and, and parasites and brain infections and liver lesions and, metal loads and, whatnot. So, all the ingredients are there. The public is essential because they're out on the bay trails and on the beaches, riding their bikes or walking, on a routine basis. And they often were kind of familiar with what's typical, unusual, noteworthy, and will see these things and then rightly report them. We're trying to get more and more people to be aware that there is a reporting network, ongoing long-term monitoring study in a lab capable of addressing these issues. So it's evidence collecting in some cases. We've had a number of big spills in San Francisco, the Cargill spill, 2006. There's always something going on, right? It's a busy bay with, shipping increases, whale stranding, discharges, accidental, whatever. There's a bunch of stuff, plenty to look after and take note of. There's a network we have established. If people see something, perhaps it's just leopard sharks and bat rays cruising by or something unusual, interesting. We had a big mola in Alameda, Maureen Anders, and a bunch of other people were looking at, not something you see every day. And there's been a lot of valuable specimens. Again, Erica Chang, and in particular Maureen Anders, over the last 4 or 5 years has collected so many valuable, unusual specimens. In some case, potentially case-sensitive specimens in case of, discharge or tide gate lockage, which, sometimes locks in a bunch of, animals which perish because of water quality diminishing. The tide gates open. Those dead carcasses, a dozen to 3-4 dozen will get discharged out with the flush tide. They'll accumulate, the deposition of carcasses over time will cause an, epizootic flourish of all these it's a great scenario if you're a microbiologist and like to study nematodes and hookworms and protozoas and carnivectors and all the stuff like that. But, it's alarming that it kills huge numbers of sharks in 2002, 2006, 2011, 2017. We had thousands of sharks washing and rays all concurrently. The sharks made the press. And, the bat rays were of equal number as well. We had, equal or greater numbers of striped bass washing up. And there's a big question, why? What's going on? With a heavy rainfall, the water was so fresh, all the animals died of happiness, it's runoff, watershed issues, and tide gate lockage, and, stagnant water discharges, and mystery goo, soils birds and stuff like that. It's a big city. You're going to have these issues. And it helps when people are at least recognize, oh, that's unusual, and can take corrective steps. We've had a lot of good, progress by some of the local public works, crews, right, that have a plan to respond. Like the town of Corte Madera isn't chronic for strandings, but they do have periodic. They have tide gates, and they do get animals locked inside that perish at times. But they're prepared for it. We've been up there a number of different times, training and reorienting and reviewing the equipment and all that, and they're ready to go. They've done a lot of good work, and that's kind of the way. The public works are alerted, prepared. The public is observing and reporting directly to myself or that public works office, whose crew can then dispatch down to whatever creek it is the report's coming from and remove animals. Oftentimes you'll have a 3-4 day window to remove and rescue animals live.
Sean,
the water
if I could,
Yep, go ahead.
Okay, so I was wondering if, so we had pulled out a short clip of a bat ray rescue, I think from one of the situations that you just described. And also, I think Maureen Anders might be in our meeting tonight. Is Maureen here? We know that Dr. Okihiro is here, and we're looking forward to hearing from him in just a few moments. So I was wondering if you could, we could get a picture of what it's like to respond to one of these situations when you know, an animal is still alive and reported.
It's a perfect example. Yeah, Maureen Anders of Alameda is a perfect example of, you know, a citizen scientist, a volunteer. She's been with us for 4 years at least. I think better part of 5 years, and she's around there all the time anyway. And she noted stuff happening in 2017. That's when we began to, you know, enlist her help as a volunteer, and got just so much done. She collected so many samples, and if she can weigh in and has anything to, you know, share with the audience, that would be a good step. You know, as well, Mark, if he's here, that's epic. He's the boss at Fish and Wildlife Department's lab. Inevitably, everything comes his way. I know he's neck deep in sea bass right now on another project, so he has to juggle this stuff. And I really can't commend him enough for not only the, you know, efficiency that he's able to do with the shark and ray stuff, but he's also multitasking with these other, you know, very, you know, important issues. So with Maureen, there's a volunteer. Mark, perhaps Erica Chang is involved. You know, our
hero. Great to see you.
She's been super helpful the last recent years. One of Mark's proteges, and he's as well got a you know, cluster of labs and people. He just kind of generated a cyclone of interest for this otherwise sort of languishing field, which is I think emerging in the last, you know, since 2011.
This is great. I'm so glad to have you all here. I'm wondering if we could take a moment to talk to Maureen. And Maureen, can you tell us a little bit about what it's like to respond and what got you involved in this organization?
Sure. First, I want to thank Sean and you, Katie, for giving me the opportunity to participate in the chat tonight. So, again, I'm Maureen Anders, and I live in Alameda, so I'm fortunate that I walk the beach most mornings, Crown Memorial Beach. And about 4 years ago, there was an article in a local paper about several leopard shark strandings in the Bay Area, and there were a couple of phone numbers to contact in the event you had seen some stranding. So I reached out and spoke with Sean, and he was very passionate, enthusiastic about what I had seen and asked if I was willing to document via photos of what I saw. So I started doing that, photos with the date, time, location of what I saw, and which, you know, various fish, leopard sharks, smoothhound sharks, an electric torpedo ray, guitarfish, striped bass, many other different types of fish. And shortly after that, Dr. Mark Okihiro with the California Department of Fish and Wildlife, senior fish pathologist, also reached out to me and wanted to receive those photos as well. So I started sending him photos, and then Mark asked me if he could send me a cooler with some trash bags and gloves, and if I could potentially collect viable specimens for him. So I agreed to do that. I think I've sent him leopard sharks, smoothhound sharks, and I know electric torpedo ray and some other fish, I believe, over the past 4 years. More recently, Erica Chang, who is they are a grad student at UC Davis and work closely with Mark. They have met me on the road. We kind of meet halfway, and I've passed off leopard sharks and I think a couple other fish to them as well for their research. Mark is extremely patient in educating me on what the specific criteria is to identify and what is he calls fresh dead specimens. So yeah, I don't always get it right, but he has a good sense of humor. So I'm grateful for that. And I've learned a lot from him. Both Sean and Mark are extremely passionate about their work, and both are more than willing to share with me their knowledge and expertise. So I've learned a lot over this past 4 years, and I'm going to continue walking the beach and documenting for them for their continued important research. Thanks.
Sounds fascinating. And I mean, is it sometimes scary? I mean, do you have to deal with jaws and you said an electric stingray?
The electric torpedo ray,
Yeah.
I was really large and I spotted it, and then Mark had asked if I collected it, and then I went back and then it was there the next day, but it was far too big for me to collect. So he had a couple of colleagues that worked for the East Bay Regional Parks, and they collected it and sent it to him. So, but it's all good.
Yeah. And welcome, Dr. Okihiro. We're really pleased to have you with us, and looking forward to all of you talking about the importance of both monitoring and lab work. I'm wondering, what's your, how do you feel the importance is of citizen scientists helping out in this awareness project, getting people to know what's going on with sharks and rays and respond appropriately?
It's extremely important because, the scientists are spread so thin, we just can't respond very quickly to most of these strandings. So, we really depend on people like Maureen walking the beach and Sean getting all of this information from people who call in, so that we can figure out which ones we can respond to, and then try and figure out the cause of these strandings. I mean, Fish and Wildlife does not have an official shark stranding program. There's no department or segment of Fish and Wildlife that responds to stranded sharks. So we're really dependent on the general public to help us out and to alert us when a shark or ray does end up on the beach.
So you alluded to a couple of things. The importance of sharks. I mean, you and Maureen too are devoting so much time and brilliance and whatever to understanding sharks. I want to say something about why you feel this is something that is possibly not neglected and something that is really important.
Well, unfortunately, most of the funding in these state and federal agencies, it's dependent on a commercial worth of a species. So abalone is a highly valued commercial and sport species, and many of the finfish species are highly prized by sport fishermen and commercial fishermen. But sharks don't rate very high on the priority list, and so that's why we're trying to pick up the slack here, because to anyone who's studied a little biology, they know that sharks as apex predators are a critical component of the ecosystem. I mean, without your top line predators, you would lose a key link in the food web. Sharks serve a purpose. They're there to take out all the weak links in the other species, and so by doing so, they're ensuring that the best genes of all their prey species are passed on, and so sharks ensure that the survival and the best prey species, whether it's seals and sea lions or finfish, that you get the best of the best that survive, because all the slow and the weak ones get picked off by sharks. So it really is critical that we, protect these apex predators and to figure out, why they're stranding.
So what are, we've heard of a couple of hypotheses. You want to just, with somebody, what are the ones that you might have in mind when you go out and make observations and do intake that sort of guide you into making sure that you collect the right information? What might be causing stranding?
Well, it's a huge variety of causes, but as we get more and more samples, we've begun to narrow down what kills different species of sharks. Great whites, for example, we've only had a handful, but so far the majority of white sharks we see on the beach are trauma victims. They get hit by boats, they get big propeller wounds, they get fishing related injuries, big hooks, or we had one recently that it died because it was trailing, a long length of fishing line, and it got tangled in a buoy, and so it drowned because it got wrapped up. But with other species, these strandings are often related to infections. Some of these are bacterial in nature. Others are caused by protozoa. And, we also have quite a few unusual cases of strandings. We've, over the last year, we've gotten several large 7-gill sharks, well over a couple 100 pounds, that have stranded. And we believe the cause is they've eaten venomous scorpion fish. So the causes are pretty wide ranging, and the only way you can figure out what's killing these sharks is by pulling them off the beach and doing a thorough necropsy. And so it's so having all those eyeballs out there, there's people walking the beach like Maureen and thousands of others, is a huge help to what we're doing.
So we'll repeat this again, but we have Sean's hotline number, definitely something that everyone should take away from this presentation. And I want to give people time to ask questions, but Dr. Okihiro, you had a presentation that you're going to share with us that looked really fascinating about lab work. Are you able to share that, or would you like us to do it?
Let's give it a try.
Okay.
Okay, can you see that? It should be a start of a PowerPoint presentation.
Yes.
Yeah, we have it. Good job.
Okay, great. So I'm just going to go over two big groups. Here's a short list of about half the species we see strand in California. And again, it's pretty much every species you find in California and in the Pacific will strand at some time. But today I'm just going to be talking about primarily bacterial infections in salmon sharks, and then this protozoa, this Miamiensis avidus, that is the primary cause of leopard shark strandings in San Francisco Bay. So these are the small salmon sharks people find on beaches, and they're often mistaken for small great white sharks. But but they're a different species, and they actually strand every year. And this phenomenon goes back, over 100 years. So this is a naturally occurring process that happens every year. But it's pretty interesting because we believe that the sharks are infected in utero, and they're actually born with the bacteria already, inside their brains. And this almost certainly happens because sharks have these two small ducts at the top of their head. And when the uterus becomes infected, the bacteria are able to gain entry into the shark via these small openings. And it's really hard to see, but there are just these two small openings at the top of the head that lead to the inner ears. And when we do the necropsies, we can culture the inner ears and the meninges of the brain and pull out this carnobacteria. Over the years, in addition to these salmon sharks, we've also seen the same infections in common thresher sharks, short fin mako sharks, and actually we did find it in one electric torpedo ray that Marine gave us. With the leopard sharks, it's a different scenario. Unlike salmon sharks, where it's an annual event, with leopard sharks, it's a much more sporadic mass mortality situation. We know that these mass mortality events go back at least 50 years, and they primarily involve leopard sharks, but not exclusively. And in San Francisco Bay, it's mainly a South Bay phenomenon. Rarely do we see these salmon sharks strand in the North Bay. And the other thing is it's a springtime phenomenon, and it's heavily correlated with heavy winter rainfall and a lot of spring runoff. So, it's June, and we're in another drought year, and so the leopard sharks, are not stranding. With the big epizootic a few years ago, we did see 3 species of sharks that were involved. In addition to leopard sharks, Marine did find a few brown smoothhound sharks that stranded. And we also saw other ray species and a few teleost finfish. This is a distribution map of where the strandings occurred. And all the red markings are leopard sharks, and the black diamonds are bat rays. So you can see that there were a lot of bat rays that stranded in 2017. And this has been consistent over the years. Whenever there's a big leopard shark die-off, often there's a similar die-off in rays. And here's our cluster around Lake Merritt. Lake Merritt had a high number of bat rays that stranded. But really, the biggest areas where we see most of the strandings is in the Foster City area, and that's what happened in 2017. And again, this shows the number of strandings per month, and you can see that it really did peak in spring. April and May, we had huge numbers of strandings, and then it dropped off rapidly. And we saw the same pattern with the bat rays. Starts off in February and March, peaks in May, and then by the end of summer, it's gone. It took us a long time to figure out the cause of these strandings. We went off in a lot of wrong directions for a while, but in 2017, we did finally figure out that it was this virulent protozoal organism, Miamiensis. It's a single cell ciliated pathogen. I actually see it a lot in hatchery white sea bass here in Southern California. And we're pretty sure that it's coming in through the nose. So the parasites are in the water. They come in through the nostrils, infect, the olfactory lamellae, and then go right into the brain. This is a necropsy shot. And the two red ovals out in front are the nose. And so that's where the parasites come in and gain entry into the brain. This one doesn't help a lot, but one thing I do as a pathologist is I do a lot of histology. So this is a fixed specimen of olfactory tissue in the nose. And when we did that, we could find parasites in some of these stranded leopard sharks. And this is a dissected brain, but most of the damage is in front in the olfactory bulbs and the olfactory lobes. We had a lot of help in 2017, and we're working with a medical student from Dr. DeRisi's lab, and she was able to confirm that this was Miamiensis avidus, this protozoa, via two molecular assays, PCR and metagenomic next generation sequencing. And right now we're just trying to figure out what are the other contributing factors to these big die-offs when we lose hundreds or thousands of sharks and rays in San Francisco Bay. We know that the sharks are naturally aggregating during these spring months. So they're in the area, they're in high density. We know that the die-offs are correlated with these heavy runoffs. So all that fresh water in the South Bay, low salinity could definitely be a stressor, but all that fresh water is also carrying a huge contaminant load. So there could also be some sort of, xenobiotic chemicals in that runoff that are, contributing to immunosuppression.
And Dr. Okihiro, we're getting some really great questions in chat. Let's see, I want to give people a chance to ask questions as well.
Okay. No,
are you going to be able to stay a little bit longer
yes.
after 8? So maybe we could return to this and give people who have to leave at the end of our program a chance to ask some questions, and then we can return. Would that make sense?
Yes, I'll be here.
Fantastic. Okay. So again, we have the chat is open, and we had a question. I can't see all of them, but we had a question from Matt asking if the parasites that you're describing can go into humans.
No, but there are similar protozoa that do attack humans. So there is a form of protozoal encephalitis that does affect humans, but fortunately, this particular species, as far as we know, has not made the jump to humans.
So people were worrying about falling into Lake Merritt.
And bat rays are still a big question. We've necropsied a few bat rays, but we have not confirmed even though we are highly suspicious that it's the same pathogen. So bat rays are something we want to look at in the future.
Okay, so I'm going to, somebody asked for the name of the reporting network, and I think we can put some of those, the Sean's hotline number, and then there's some other other numbers that were shared with us from the department and Fish and Wildlife, and also iNaturalist has a place where you can photograph animals or stranded sharks and post them, and so they could be viewed by somebody interested in that.
Katie, I have the stranded sharks and fish hotline up on the screen right now.
Great. Okay, and I have another one on a slide that I have prepared, but we don't want to go there right now. But I will put it in, and I will send it to everybody in a post chat email. We'll send these links to you. But here's a good place to start. Yes.
Are we interested in any more questions from chat?
Very much so. So just in mechanics wise, I think we should take questions, and then we'll have a few minutes of wrap up at 8 o'clock, and then we'll reopen for more presentation and questions. Go ahead, David.
Well, I see a question from Matt Baker. Can those parasites go into humans? And it's a follow-up question from Joy Harnett asking if someone is boating on Lake Merritt and capsizes, are they likely to get the pathogen that the sharks take in through their nostrils? That would be through the human nostrils. Will people get that in their nostrils also, I see? It seems very deadly, is how the question ends.
As far as we know, this parasite is not transmissible to humans. The protozoal encephalitis that affects people is a different species and is, I think, associated with stagnant fresh water. But as far as I know, go ahead.
Yes, I have a question in the same vein. Well, it's regarding the leopard shark, though. If the parasite is situated in the brain, is it key to the disease and the stranding? I was curious as to how fresh water or from heavy rainfall was affecting that. It would seem as if whether there was heavy rainfall or not, the parasite would still trigger the strandings.
Well, we think it's possibly a stressor. So the sharks are there, and during heavy runoff years, if you have a lot of fresh water coming into the South Bay, then the salinity is going to drop sharply, and that might be enough of an immune suppressor that it allows more sharks to get infected. But really, we'd also like to find out what's the natural host for this parasite. My suspicion is that it's a benign parasite on some other species in San Francisco Bay, and that only under these certain, years when there's a lot of runoff, this parasite is somehow able to amplify, multiply, and then make that jump to leopard sharks. We have the same problem with white sea bass in Southern California, where we get these periodic big infections where thousands of fish are lost, and we just don't know which species out there is harboring this virus. Is harboring this protozoa. The thing with parasites is that if a parasite is well adapted to a host, then it doesn't kill them, right? It's not any benefit to the parasite if you kill your host. It's the way they evolve is that it's either mutually beneficial or the parasite doesn't do enough damage that its host dies, because again, there's no benefit to the parasite that way. So that's why we think the leopard sharks are an unnatural host for this Miamiensis protozoa.
Thank you.
I have a question that was sent to me by email. There was a lot of discussion about in 2017 about rays and sharks being trapped behind tide gates and other kinds of infrastructure. At Lake Merritt, we have a trash grating that that filters the water coming in and out through the channel with a distance of about 9 inches, I believe, between the slat, no, 4 inches, I believe, between the slats at the top, and then it goes, there's a larger, how have I got this right now? There's a larger distance at the bottom of the channel as the water flows in and out, so that fish, hopefully the fish would be able to go through the larger aperture if they weren't able to go through the top one. Do you think it's worthwhile to review the escape routes for larger fish on some of these structures that we have?
Well, you know, it's a dilemma, right? Because if you increase the screening to Lake Merritt through these intake pipes, then it becomes a maintenance problem, because the smaller the openings are, the more fish you exclude, but then the faster they foul, and so there's less flow of water into Lake Merritt, and the water quality goes down. So we see the same problems with the net pens we use for white sea bass. You know, if you know, you want to have the largest screen size you can to increase the water flow, and still exclude predators.
So in Lake Merritt, they're excluding large of debris and trash, so that they don't get into the pump system, which is, you know, an expensive system that is used for flood control. And actually, the trash gratings are cleaned off, I believe, at least twice a year now by divers, to keep the marine growth from blocking the flow of water in and out.
How do bat rays get into Lake Merritt?
Pardon?
How do the bat rays get into Lake Merritt?
They do. They have to go through the tide, the tide channel. They have to go through the tide gratings in order to get in. There's no other way. So, and they're there all times of year. We have reported bat rays in the lake, but more often in the channel, past on the lake side of the tide, the flood control station where they have these gratings. So they get in and they get out, and we haven't seen the die-offs like we had seen in 2017 prior to that, where we had a large number of bat rays dying within a period of a month. That's not a usual thing. So
it may be that under certain circumstances you get stagnant water conditions in Lake Merritt, and if you had that, then you would give the protozoa a chance to amplify, so you'd have a bigger cloud of parasites, and it would be easier for the parasite to jump from ray to ray. In 2011, we saw a big jump in leopard shark and bat rays that died in Redwood Shores. And Redwood Shores also has a lagoon system where rays and leopard sharks are trapped within a body of water that's regulated by tide gates. And in that die-off, it started in Redwood Shores Lagoon, which is south of Foster City. But then subsequently, expanded out into the entire South Bay. But these bodies of water, these small bodies of water like Lake Merritt and Redwood Shores Lagoon, you know, certainly could make it easier for the parasite to multiply and therefore infect sharks and rays.
Thank you. So we are up on 8 o'clock, and we'd like to take a few minutes to give our end of program closing, and then we'll reopen for more discussion and questions, and we can hear more of Dr. Okihiro's slideshow. It was fascinating. So if everybody could bear with us, we're going to open up and tell you about some of our upcoming events. And let's see. Here we go. So this is Lakeside Chat series, and we're so excited to have wonderful speakers on a variety of topics. It's planned for the rest of the year, really. Next month, Damon Tai, local naturalist and fungus and fungi lover, is going to talk to us about the mushrooms of Lake Merritt, and that'll be a wonderful presentation. In August, we're going to hear from Susan Schwarzenberg and some of her colleagues from the Exploratorium, and talking about the place of a museum and observatory in the urban setting and connecting people with the environment that they influence and they're influenced by. So she'll be talking in August. So here's the slide for Damon's talk coming up. I've gone on many of his walks and heard many of his talks. This is going to be really amazing, and I encourage everyone to sign up for that. We'd like to thank our producer, Rob Lamone, for media and videos, all of our Rotary Nature Center Friends crew, who's been helpful in many ways, Patty and Kirsten and Janice and Betsy, helping to behind the scenes with the program, with the trivia. Also, we are doing things in real life at Lake Merritt, and we're very soon going to be having a monthly Adopt-a-Spot session, cleaning up around the Rotary Nature Center grounds, and also doing some work at the restoration marsh that's on the shores of Lake Merritt, between the boathouse and the nature center. We're planting native plants and learning a bit about restoration processes and education. We want to thank the Frederick E. Hart Foundation for support for our programs. Lake Merritt Institute has also contributed, with many people who have been involved in our activities and processes over the past 2 years, supporting the idea of an interpretive services at Lake Merritt for the people of Oakland of all ages and backgrounds. And so we are a citizens group, and you can go to our website and find the YouTube links to all of our programs and see some photos of some of the educational activities and stewardship activities that we do. So we'd like to thank so many people. We have been involved in a variety of activities and hope to do more in the coming year, as we all are able to spend time in real life at Lake Merritt and out in nature. And that's it. Okay, so I will unshare.
And thank you, Katie. Thank you, Katie. And thank you, David, for making this happen.
And thank our audience for being here. And this is really great. So we ended really pretty close to on time.
I want to say thank you, but I'm concerned about my volume. Am I still having a heavy echo out there?
Yes.
I'm just waving my thanks.
A teens. Yeah. Okay. Let's see. So now we're going to open it up for more discussion and questions. So do we have people in? I can't really see all of the chat, so I'm going to ask the chat wranglers and anybody else. At this point, you can unmute yourself, raise your hand, and ask a question. So I have a question about plastics. So here at Lake Merritt, we're really worried about and we're actually under a mandate to reduce our plastic flow out into the bay to 0 over the 2009 level in, I think, 2022. So we're under pressure. What kinds of impacts are there on sharks and rays on all of this trash that's going out into the bay? Sean, we lose Sean? No, we didn't lose Sean. Please unmute yourself. By the way, where are you, Sean? So I saw, I wanted to tell everybody also, and we'll send this out, that there are just a myriad of wonderful videos where you could see the Pacific Pelagic Shark Research Foundation responding to issues. And, so you can see the context and what they do and everything. It's really great. I'll share that with everybody in a post chat email. But what about plastics, microplastics? Are there any things that you have noticed about them in terms of the health of sharks and rays?
I think Sean is still muted.
Oh, okay. I'm going to ask him to unmute. Can we, Rob? Do people have to unmute themselves? Okay. I will send him a message.
There he goes.
Oh, good. Okay. So, Sean, I was asking both of you, have you seen any effect on sharks and rays of the trash that emanates from Lake Merritt and other parts of Oakland?
Yeah, I have, not in Lake Merritt per se, but I can say that monofilament fishing line is the most obvious marine debris issue. That's plastics. Regarding sharks, both accidental, sharks get into mischief, try to steal people's, they catch a fish, they're trying to reel it in, and the shark will intercept that. As well, there's quite a few people, and apparently a growing number of folks targeting sharks for amusement and or actually illegal sport fishing for, like, say, white sharks. As Mark presented a good example where a shark trailing monofilament line, we can't figure out whether that was deliberate or not, but often enough the sharks will end up trailing fishing leaders. Oftentimes it's pretty heavy gauged, people intending to catch something big and heavy like a shark with teeth, and that will and has resulted in entanglement issues. Somewhat different, but similar, to whale entanglement issues as well. We have had at least 4 different sharks reported in Monterey Bay trailing rope, actual rope, some of it polypropylene plastic rope, 3 in Shark Park, 1 of which we were able to remove, and then several animals we've been able to, just by looping the fishing line, trailing fishing line around our GoPro, pop at least a segment of that line off, because that poses a hazard. And then I have over time, like during, when we were blue shark sampling, when blue sharks were more abundant, we've collected and removed plastic packing rings, which marine mammals and sharks and fish will often enough somehow figure out a way to flip that over their head, they're investigating it or whatnot, and they can't go backwards, and that remains stuck on their head. And if it's a younger animal, it'll continue to grow into that with that ligature, stuck around their head and around the gills, and that can be terminal. And then ingestion issues, to my knowledge, Mark maybe have more examples of that, but they're very good at disgorging stomach contents. They throw up a lot of bones, teeth, stingray stingers, debris that they will consume, digest all the optimal nutrients out of, and then just disgorge all of that. They actually can invert their stomachs, and discard a lot of stuff, and then slurp their stomachs back in. So aside from hooks, stingrays, scorpion fish, it's a hazardous, menu out there for these sharks. They have a challenging, job description as it is. And, so there are natural causes, that affect a lot of these, a notable amount of these stranding events. Plastics is there. It doesn't pack the gut, so far as I know, like say seabirds, sea turtles, marine mammals and stuff, which aren't able to, to the same degree disgorge, literally their stomachs out of their mouths, and then slurp it back in. But the entanglement issue, the abundance of plastics, discarded, broken, lost fishing line, 6-pack rings, it messes up a lot of stuff. The birds, the turtles, the fish, sharks, and whatnot. The marine mammals. They don't get the memo. They're smart, but they're not that clever or perceptive, and they're very curious. And so yeah, that is a major issue, worldwide, and we do see it here locally.
That's interesting.
A note about the tide gates. Mark made some good points there. Not all of that is entirely figured out. Environmental engineers, they're calling for you to look at tide gates and try to figure out how do you sift out debris that would otherwise be incoming into a lot of these urban, suburban waterways, lagoons. Some of them double as stormwater basins. How do you rectify that with water control? I remember 15 years ago, sea level rise was the debate. It's not anymore. It's the fact of life. So a lot of these, infrastructures in terms of tide gates, pump stations, etc., they're varied, and the ones in Corte Madera are going to be different than the ones in, say, Redwood City or Foster City and Millbrae, and most of the former tidal marine estuary environment is now, kind of engulfed and converted into these urban suburban waterways, and in most cases they're quite nice, but you do have these complications. The tide gates aren't always closed or open, so regardless of how wide the grading is, etc., some of them are just tubes that get fouled with mussels and stuff like that. That's a complicated issue, and I've talked to a lot of people about it, and I can't figure it out. It's going to take someone with engineering understanding and a complementary concurrent understanding of these environmental issues, which are still pretty obscure. Some of the stuff Mark's done has been fabulous for stimulating curiosity, interest. It's never good news, right, when it shows up in the newspapers and kind of stuff like that, but that's the only venue really for people even being aware of it in large part. But this will kind of help. So tide gates are an issue. What I've noticed, going back to 2002, is that when the tide gates are closed to avoid flooding. So at low tide, they'll close the gates when the water levels are low, and that will accommodate the rainfall anticipated. Oftentimes, that's going to change the pH, everything, oxygen levels, freshwater influx, and everything. It'll deteriorate quickly. You have a 3-4 day window while those gates are closed when you notice animals in there to remove them. Corte Madera has done a really good example. It's tough. They don't get them all, and you're always going to have some mortality. I don't know how to rectify that, really. Got to be pragmatic and realistic about it. But a big part of that is having public works crews alert and ready and aware of that, even as an issue, so that they can, when they get reports from myself or the public at large, property managers, etc., hey, we got a bunch of bat rays here that have been dead for a week and they really smell, right? Or you have people more alert and watching, oh, there's bat rays here, they're swimming around. That's your cue to go get them or remove them. Corte Madera has done that. Some other North Bay people have been, more prone to that. It's not as much an issue as Mark says, as in the south end of the bay. Also keep in mind, not all of the bay, the South Bay, etc., is accessible through bay trails. They have a huge swath. I invite everyone right now to go to Google Earth and look at the satellite images of San Francisco Bay at large, and in particular, Redwood City. San Mateo is an area and it's one of the nicest areas around, right? You know, and including their waterways, but those do double at times as stormwater basins. Animals get caught behind the tide gates, can die off in their dozens. Those, when the gates reopen, get flushed out into the near shore bay trail area, right? That deposition, accumulation of rotted carcasses may be a vector for these plumes of protozoas and other stuff like that. And we're looking into that, you know, with the carnobacterial issue. You know, something that caught my attention early on is the first detection of a carnobacterial die-off was in a fish farm, right? With salmon farming. And, you know, things you learn as you go. Carnobacteriums are actually produced in a lab nowadays and used as an additive preservative for not only fish farm foods, but produce, meat, markets, because it eats listeria, and carnobacteria is a predator of bacteria, the stuff that stimulates spoilage. And so if you know, put that as an additive, it can slow down, you know, spoilage and waste. But, you know, there's a concern there. Maybe this fish farm lab produced variant has mutated or something. I've heard that. It's a bit far-fetched. It's way too early to speculate that, but it's on the radar for sure. And people like Mark will be able to, you know, kind of dice that up and tease out what information is available.
Definitely,
And,
a really
you know,
complicated.
it could have very
So Sean,
long-term.
we've got a couple of specific questions here. Yep. Coming up. One is, how much do leopard sharks and bat ray populations move around the bay during the year? Are they
quite a bit?
Okay.
Yeah, they are migratory. Yeah, actually, you know, the geneticists again, the lab people have been able to discern several distinct genetic groups, not clades per se, but, you know, genetic signatures among the leopard shark population. So, you know, the sharks we have in La Jolla are going to be noticeably, you know, distinct from those in San Francisco or further up the coast or down in Baja, you know, etc. And, you know, maybe the case, this needs, you know, bearing out in further research, but, you know, we may have lost a lot of the estuarine, you know, specific leopard sharks. And so you'll have a lot of influx within San Francisco Bay of migratory leopard sharks, as well as long-term ground species leopard sharks. Leopard sharks are seen out of the Farallon Islands, you know, at times as well. So they're not strictly coastal, they're on the shelf. But there's a lot of variation among leopard sharks. Sometimes you can tell by this leopard spotting patterns, you know, as well.
Okay, Sean,
you know, they're not considered endangered at this point.
Some people waiting here to ask a question and follow up here. Janice, do you have a question? And then Justin, and then
my question is with the microorganism. There was one that you used the term Miami, and I can't remember. I didn't write down the rest of it. So it was Miami. Do you know what I'm talking about?
Mianensis avidus.
Oh, Mianese. And now Mianese is. No,
I never heard it.
Is that only from this area, or is this happening to other bat rays and leopard sharks in another region?
Good question. Yeah. Mark could probably give you a more thorough answer, but so far as I know, it was first detected on the East Coast and then subsequently, surprisingly, found here in Monterey Bay. I don't know if it's an invasive species or something that we just didn't notice before. Part of it is that, you know, until you look, you'll not notice it. You know, and these sharks and fish have largely gone, you know, neglected for decades in terms of, you know, most of the focus was elsewhere, and for good reason. You know, marine mammals, marine birds, and slowly the sharks and the fish thing have become more obvious and of interest as the field expands. And so we have much more, you know, many more students, and it's not just a you know, objective biology question or fisheries biologist, you know, focus, but, you know, environmental studies and you know, and all these other fields, you know, veterinary medicine, which overlaps with it. And you know, that's my understanding.
Okay, can we ask?
Say again, Katie.
I'm sorry to interrupt you. We have a couple people wanting to ask questions before the end of our time. So I wonder if you have a moment here to, Jonathan, would you like to ask your question?
Yeah. Hi. This is specifically for Dr. Okahiro. We've talked a few times, but I'm a grad student down at UC Merced. And I've been wanting to bring some data up to Dr. Okahiro that I've been doing some habitat modeling with the leopard shark population in San Francisco Bay and looking at how salinity and temperature and a few of these other abiotic factors are affecting it. And I'm seeing that leopard sharks in the bay their preference of habitat is dropping right around 16 parts per thousand in salinity. And just wondering if a lot of these like die-offs are happening below that threshold, or if we know like what salinity threshold we see a lot of these die-offs at.
No, we don't have that specific data. We just know that when we're chronicling these big die-offs, you know, in 2006, 2011, 2017, that they correlate very well with, you know, wet winters and heavy spring runoffs. So it's just supposition on our part that the low salinity is possibly a stressor that's helping to increase the number of sharks affected and the severity of infections. But again, it could also be that these heavy runoff years also mean there's more contaminants being washed into the South Bay, and that it's, you know, an herbicide or a fungicide, something that's coming in with this runoff that's causing immunosuppression. So, you know, really, the people at UC San Francisco have been able to culture this pathogen, this Miamiensis protozoa. And so really what you could do is run laboratory experiments to, you have the pathogen, you could run controlled experiments where you did stress them with low salinity or selective pesticides to see if you could reproduce the disease. But right now, this correlation is just really general, but it is a pattern that's holding.
Cool. Definitely. Thank you very much. And I'll have to email you again soon.
Okay.
Yeah, very interesting.
I had some to add. That was a good question. One thing to keep in mind in regards to San Francisco's leopard sharks and bat rays is they do carry a high load of contaminants already. You know, so when they're healthy and robust appearing, when you open them, they have liver lesions and a lot of other stuff that's a bit abnormal. And, you know, since 2011, they've been declared by the public health and safety people as unsafe to eat due to that heavy load of toxins. And so that should be considered as well. These sharks are already sort of weakened just by the ambient bioaccumulative toxins they've gathered on. And then, you know, concurrently and additionally, there are a large number of these, you know, pathogens, including parasites, you know, and whatnot that, you know, may tax them. You know, definitely water, you know, watershed issues, runoff is a big component of that. I think anytime you have a big influx of anything, as Mark pointed out, when you do have rainfall, it's not because the water's too fresh. You know, salinity is low. We need to remember leopard sharks and bat rays in particular are very well adapted to low salinity and high salinity, low oxygen, and fluctuating temperatures. It's part of life in an estuary for these sharks. So typically they're the last thing to die off, and when they die off, you know, that's a concern. The other thing I wanted to point out before I forgot is a lot of these strandings in the spring involve adult females with a cargo of over 30 young. So it's not just one shark we're losing during these big springtime, events where, they're epizootic die-offs, but we're losing young of the year, springtime, animals, is what. So yeah, of course, they're marine animals, but they're very well able to cope with low and high salinity. And I think while the heavy rainfalls are, is certainly a component of that, you need to keep in focus. These sharks are already compromised quite a bit by mercury, and PCBs and all the various pathogens and toxins. And when there is an accumulation and deposition of carcasses in an area, coming out of the tide gates, for example, that's going to cause, that's a vector. It's going to cause a bloom of all these protozoa, bacteria, and whatnot. And we can't lose sight of that. Going back to 2002, I've seen numerous events. They've all started behind tide gates. Of course, that's where a lot of the backfill is going to be fresh water, and you'll have that low salinity, low oxygen, and all these other, very challenging, problems for a shark that, are rare fish that's stuck behind a tide gate. And then when they die, the tide gates open up, it flushes out and builds up over time. And these events have run all the way through August. So it does taper off, but they persist. There's some overlap that's maybe coincidental with, like, the big 7-gill sharks and whatnot. It's early in the study since 2011. Paula Schaefer published the first paper on the salmon shark strandings back in 2013 with the help of initially Dave Casper, Guy Alder, and Corinne Davis, again of UC Davis and UCSC. And then Mark came in 2011, has been kind of the boss, and kind of took all that, what little information there was available and put it together. And he's, perfect for that job. He's objective. He'll provide us with all the latest information that we can confirm, but there's still a huge amount that's not fully diced out or, ground proof yet. So we're looking into that. And, I don't want it to be perceived as like, oh, it's going to rain and we're going to have thousands of sharks die. That's not normal. And in part, that's because of loss of tidal estuarine, habitat, and maybe complications, I'm certain, with the tide gates. Some are better than others. And I felt that was, important to add to that very good question and comment.
So many questions. Do we have other people with hands up in our audience? I'm wondering how sharks are dealing with other kinds of non-living contaminants. Is that another stressor, or is there any do some of them get stirred up at certain times or in certain non-drought years?
Yeah,
affecting things.
We're looking for, everything from, radioactivity. 2011 was the West Pacific had a big disaster. So we looked at that, looked for pesticides, and a lot of these factors overlap. So Miami Ensis avidus was like, wow, look at that. We, got an ID on this. And there's still a lot of other stuff that's still being looked into, including fungal inclusions. And I think when, we have big runoff, situations, you're going to have some bad plumage as a consequence. Some areas, again, are better than others. When it rains heavily, especially after, a dry, hot summer, you're going to get motor oil, brake dust, snail killer, lawn fertilizer, all that stuff is going to go into the watersheds and pool up in those stormwater basins. So it's not just a lack of, salinity. As well, when it gets later in the summer, the rains are done, those same water, stormwater basins will evaporate. The water becomes high salinity. And again, sharks and rays are very good at these. Sharks and rays, leopard sharks, bat rays, smoothhounds, are very, very able to cope with those conditions. but under normal natural conditions, they'd be able to egress and escape. But they run into these barriers, and we have 3-4 days, you know, public works people or volunteer groups be able to make the transit, you know, up to, you know, remove the animals while they're still alive. I think the deposition and accumulation of rotted carcasses outside these certain tide gates, you know, is probably a part of this epizootic dynamic. You know, there are all kinds of things. Harbors sometimes have big bunches of anchovies move in there. They'll deplete the oxygen and die en masse. You know, artificial waterways can be complicated. Anyone who have had a home aquarium, a saltwater aquarium especially, you know how tricky that can be to keep everything just in perfect pitch, you know, in terms of temperature, oxygenation, and all that. And a lot of these stormwater basins in lagoons, you know, when they're not suited to ducks and that kind of stuff, you know, they run into, you know, complicated times. And a big part of it, I think, is getting the public works people aware and involved, you know, and on the same page too, right? It's not a blame game. No one's really to blame. No one knew until recently, thanks to Mark's lab work. And now we can look out for it, you know, with the public support and, you know, eyes looking and monitoring, interface and connection with the public works guys who are typically pretty, you know, let them fish and surf and do stuff similar on their free time. You know, they're happy to go down when they have the free time to go and scoop out animals on a certain day. You know, the aftermath is never fun. When you got a big, you know, several big dead bat rays smelling up one of the ponds near Oracle. You know, we've had that happen a number of different times, etc. So I think that's an improvement over time. We got more and more people reporting these events, and it just helps with the coordination, with the collection of evidence, you know, specimens that Mark and his colleagues, Erica,
and you, Sean, you know,
on and identify what we can.
You know, all of your work since 1990 and your dedication to monitoring and so forth, we're really at the beginning of understanding how ecosystems around urbanized area where people are affecting things, and just what happens with the natural cycle of things, how that's going to affect populations and different species. We could go on for quite a while. Some great questions. I'd like to invite everybody who's here. If you have additional questions, you can send them to me or quickly type them into chat, and we will try to get them answered in the next few weeks, or next week rather, with a follow-up email to everybody. And I think we're going to need to close out in about a minute or two, but I wanted to thank Sean and Dr. Okihiro for a fantastic presentation. So much to learn. Really, thank you so much for sharing this with us, because, you know, it's really not on everybody's radar, and it's really great to be able to share that with our audience here at the Lakeside.
Thank you so much, Katie. Good job, Lake Merritt Institute. And Mark, thank you so much. You're awesome.
You are awesome.
Maureen, good job. You're always there. Thank you, guys. Good job.
Thank you so much. It was a wonderful presentation. I really appreciate that. I encourage everybody to go to Sean's website, and of course Dr. Okihiro's website, the lab website of the Department of Fish and Wildlife. They have videos and photos and lots of information. If you have a chance to go look at it, it's really fascinating. Okay, I think
look
we're
us up on YouTube.
Yeah, oh, absolutely. So we will send, and you can help me out, Sean and Dr. Okihiro. You can send me those links. I have a lot of them, but we'll get something out to everybody so they can take a close look at whatever specifically interests them. Okay. Thank you again so much for spending time with us. It's been
outstanding. Thank you, guys.
Thank you. All right. Take care. We'll get the YouTube link out too. Okay. Good night. Night, everyone.
Good night.
Good night. Thanks so much.